Hello to our friends joining us via recording. We are wrapping up our discussion of lesson number 7, which is about the integumentary system. Remember from our class on Monday that when we talk about the integumentary system, when we talk about the skin, there are 3 kinds of skin cancer that start in the epidermis that can give us trouble. When I'm looking at these 3 kinds of cancer, and let me number them so it's easier for you all to talk back to me. Let's say that squamous cell is number 1, basal cell number 2, melanoma number 3. Is it 1, 2, or 3 that's the most common kind of skin cancer? Do we remember? Was it 1, 2, or 3 that is the most common? Yeah, several of us are chiming in correctly that basal cell carcinoma is the most common kind of skin cancer. Remember with basal cell carcinoma that the cancerous cells that are making this tumor that we see here on the skin, those started in stratum basal. So they started in the keratinocytes that are down there. Remember that when those cells become cancerous, they start to grow down in this uniform pattern. So the fact that basal cell carcinomas grow down in a uniform pattern is good for you because it means it's easier for us to remove the cancer cells. When we talk about our neighboring cells over here, our squamous cell carcinoma, we talked about how this one does not grow down in a uniform pattern. So we don't see this bottom part like this. Instead, we see these little bunches of cells that are growing all at their own rate. When we talk about squamous cell carcinoma, this one didn't start in stratum basal. Do we remember from class on Monday, where does squamous cell carcinoma start? Which stratum does this one come from? And you can totally abbreviate and not type its whole name. I know it's not English. I have 1 vote. Anyone else want to toss their hat into the ring? We're nervous about this question, I take. When we talk about squamous cell carcinoma, if we look over at my picture here of the epidermis, squamous cell carcinoma is starting in the cells that are here, in the region known as stratum spinosum. So these cells that live here in stratum spinosum are really flat. Squamous is their shape. Squamous cell carcinoma when a cancer cell starts in stratum spinosum. Basal cell carcinoma when a cancer starts in stratum basal. There's actually more than one kind of cell that lives in stratum basal though. The other kind of cell that lives in stratum basal are those melanocytes. And remind me in the chat, melanocytes make that protein melanin. What does melanin protect you from? What does melanin protect us from? Why do we like having melanin? Exactly. Yeah, melanin is a protein that absorbs UV radiation. So we like having melanin in our skin to make sure that we don't get skin cancer, hopefully. But when my melanin producing cells, when my melanocytes have their own DNA mutated, they can turn into cancer as well. And so when we look at a melanoma, like we see over here, this kind of cancer is highly pigmented because all of the cells that are making up my tumor are still making the protein melanin. They're still building this dark colored pigment. So a melanoma is going to be that dark colored pigment. It's going to be another kind of skin cancer that grows down not at a regular edge, but at these little line edges. It's harder for us to excise it all. And also, when we talk about melanoma, it's very easy for that to spread, to do something that I'll type in the chat here called metastasis, when it spreads from one part of your body to another. So when we're talking about melanoma, you can stage melanoma based on how far it has metastasized or how far it has traveled. So best case scenario, if you have a melanoma, would be that it's a stage 0 melanoma. In a stage 0 melanoma, my cancerous cells, my melanocytes, are only in the epidermis. They're only in the area that they're supposed to be. That's why I'd call it a stage 0 melanoma. Once they spread into the papillary layer of the dermis up here, then we become a stage 1 melanoma. So a stage 1 melanoma has started to go into the dermis in that upper papillary layer. If that melanoma keeps growing and it's down into the reticular layer of the dermis, that's a stage 2 melanoma. If we grow all the way down into the hypodermis, we're now a stage 3 melanoma. And stage 3 melanoma is a bad turning point for us. Because see, the problem is down here in the hypodermis layer of the skin, we also have these things called lymph nodes. So when you've got a sore throat and you go to the doctor and they feel your neck, they're feeling your lymph nodes. You have lymph nodes all over your body. It's kind of like immune system centers throughout your body. If a cancerous cell, if a melanocyte gets into one of these lymph nodes, it's basically on the superhighway to go anywhere in your body. So when we get to a stage 3 melanoma, we have the potential to go anywhere in the body. And a stage 4 melanoma, not only do we have the potential to go anywhere, we actually have gone places. So in a stage 4 melanoma, maybe the original melanoma tumor started off on your arm. You might see another melanoma on your legs, for example. Or inside the body, we sometimes see melanomas that grow in the liver or the lungs. Tumors that are a melanoma in the liver or the lungs, these kinds of tumors still keep making melanin. So if they were to be removed through a surgery, they would still have that melanin pigment inside of them. So once a melanocyte, always a melanocyte, even if we're making a cancer that goes to different places in the body. So stage 0 up to stage 4. The way that we stage a melanoma is how far it's spread, how far down or up we find it in the skin. How do we feel about stages of melanoma? Thumbs up or any questions before we move on? Angelita's good, getting some thumbs up. Perfect. I had a student this morning that made the accurate connection. This is actually a lot like our degrees of burns, right? Where it came down to how far in the skin did we damage stuff. It's exactly the same way when we're staging melanoma. How far down have these cancer cells gone? And again, it gets real dicey when we get to the lymph nodes, because once we get there, we're out of the skin. We're going other places in the body. Yeah, so Lisa's mentioning that her dad has had skin cancer. Yeah, which kind of skin cancer did he have? You mentioned it was on his nose. I'm not sure. Yeah, I think pretty typically the nose is where we see a lot of basal cell carcinomas. That's pretty common, a common location for basal cells. But yeah, not a melanoma. That's good. Melanoma, I think we mentioned this last time, melanoma by far the deadliest kind of skin cancer. So definitely don't want a melanoma anywhere, for sure. Let's see, which are the ones that present as flaky skin? Which skin cancer? Is that what you're asking, Jessica? Yes. So here I'll bounce back to a cartoon for you. When we're looking at the two kinds of skin cancer, basal cells tend to be more smooth on the surface. Squamous cell is what we call ulcerated. So the way that it grows looks kind of nasty and kind of falls apart a little bit. So I'm not sure if maybe that's what you're thinking of, Jessica, with the more ulcerated, ugly looking one. I'll bounce back to our actual pictures. This over here is a very typical basal cell carcinoma where it's got this round, shiny surface. Whereas over here in the picture that I see, this is a more typical squamous cell carcinoma. So basal cell first grows up and then grows down slowly in a uniform pattern, whereas squamous cell looks a lot uglier and grows down in those little uneven ways. Yeah, so it might have crusting, like Jessica was asking. Absolutely. Yeah, so she's saying that a doctor says when you're checking for skin cancer, looking for spots that are dry and crusty. Yeah, absolutely. I am not a dermatologist. I did tell you while I worked for one. We did a lot of basal cells because basal cells are very common. Squamous cells being the next most common. Yeah, and Cassidy's mentioning that her dad has had a lot of squamous cells. So various types of skin cancer, both of these much better than melanoma, but no kind of skin cancer is ideal. We do not desire skin cancer. All right, so when we talk about the difference between a stage 0 and a stage 1 melanoma, stage 0 stays in the epidermis, stage 1 goes down into the papillary layer of the dermis. Based on that, that division, that's what separates epidermis and dermis. And if you remember from lab, the place where the epidermis and the dermis meet each other goes up and down in this wavy pattern. So the dermis and the epidermis don't just lock together in a flat surface. They have these little things that we talked about in lab called the dermal papillae, places where the dermis reaches up. And on top of the dermal papillae, we have places where the epidermis reaches down. So the dermal papillae and the epidermal ridges, that they're called, come together to make this wavy pattern where our tissues interact with each other. That's really good for you because then if our tissues are pulled back and forth, they're anchored together much more strongly. In other words, they're able to resist friction, which is something that we're seeing down here at the bottom. The fancy anatomy word for your fingerprints is we call them friction ridges. So friction ridges, those fingerprints that you have, the pattern of every single person's fingerprints is different. Even identical twins don't have identical patterns to their fingerprints. And the reason for that is because your fingerprint starts at this dermis level. So it starts at the lower level in the skin. This interaction between the dermis and the epidermis starts in the first trimester of fetal development. And scientists don't fully understand yet what specific chemicals tell me, oh, I'm going to end up with an arch, or I'm going to end up with a loop or a whorl. We don't know exactly how it happens, but we do know that it comes from those dermal papillae that are reaching up that cause the epidermis to shift a little higher. Now, here's a question that I want us to consider. When you take your fingerprint for an art project, for a new license for your job, when you do your fingerprint and you stick it on stamp and you put it on paper, what you're seeing is the pattern of your epidermis, the skin there on the outside. But remember I told you stratum corneum, this outside layer of the skin, stratum corneum is constantly falling off. And it's constantly, it's completely dead. It's constantly being changed. But your fingerprint is the same for your entire life. Why doesn't your fingerprint change? Even though we're losing skin cells all the time, why doesn't our fingerprint change? Either I've got some serious lag, or about 2/3 of us are asleep, which I would understand. Yeah, so Cassidy and Jessica are honing into what we mentioned, that your fingerprint pattern starts not because of the epidermis, not the way the epidermis is built, but because of the dermis layer of the skin. And we're totally right. In the chat, we're mentioning things like your DNA. Your DNA does dictate some of the factors that give us some of this pattern as well. So your DNA doesn't change. And what else doesn't change is the way that your dermis is built. My epidermis changes all the time. I'm constantly shedding off skin cells. That happens all the time. What doesn't happen all the time is my dermis changing its shape. My dermis is always the same shape. Back in the day, Dr. Ollis had a student who I think was probably a pyro. He really liked fire. So he asked me if I wanted to burn off my fingerprints, what degree burn would I need to do on my fingers? Number one, ouch. Why would we want to do this? But here's my question I'm going to pose to you all. What degree burn do you think we'd have to do to change our fingerprints? What do we think? Yeah, so we're pretty divided between 2nd degree and 3rd degree. So we all recognize that a 1st degree burn's not going to do it. Remember, 1st degree burns just stay in the epidermis layer. In a 2nd degree burn, remember that what we talked about is we make blisters because I fry some of the proteins that are found up here that attach these two things together. But before I actually destroy the tissue in the papillary layer of my dermis, I have to go even deeper and damage even deeper in the skin to do that. So when we talk about what degree burn we would need to do to change our fingerprint, yeah, it's going to have to be a 3rd degree burn. One of my students this morning said, you better do a 3rd degree burn to be safe because of the true crime TV shows. I counter that with saying, just wear gloves, right? You didn't hear this from Dr. Ollis, but just wear gloves if we're going to commit a crime, because that's way better than going through a 3rd degree burn on your fingertips. So if we wanted to get rid of this pattern, we would have to get rid of our dermal papillae, and that would be incredibly painful. Probably not worth it. Checking out my chat here. Following the waves. Yeah, just like the different waves if we were cutting grass. Yeah, the patterns of your fingerprint. I always love when I put this up in class, because as soon as I put up the pictures of the fingerprints, everyone starts staring at their fingers. And I totally get it. I don't know what my fingerprints are until I look at them too. So these different patterns coming from deep inside your dermis, leading us to these different epidermal ridges that we see. So here's what we've talked about so far. We've talked about the epidermis with all of its different cells, that it's waterproofed, it's really tough. We've talked about how the papillary layer of the dermis goes up and down and helps to create our fingerprints, or our friction ridges. But the other thing that's really important about this papillary layer of the dermis, and the reticular layer for that matter as well, is that these are places where we have a lot of blood supply in the skin. So let me put a word in the chat for us here that's new. That word is avascular. Avascular means no blood vessels. When I talk about the epidermis layer of the skin, by itself, we would say that this layer is avascular. It has no blood supply. There's a couple of things that's inside your blood that cells everywhere in your body want. Can we think of what those things might be that the cells everywhere in your body want to have? What do we store inside or transport inside blood to the rest of the body? Yeah, several of us are remembering my cell's favorite food. That's the glucose. Definitely find that inside blood. And I also find, as a couple of us mentioned, the oxygen too. So my epidermis doesn't have any glucose or oxygen on its own, The papillary layer of the dermis has little blood vessels right up next to the epidermis. And when we look in the reticular layer of the dermis, there's even more blood vessels down here. So primary function of these blood vessels is to feed the epidermis, to give it the glucose and the oxygen that it needs. But the other thing that those blood vessels in the epidermis help us to do is to maintain our body temperature. So we're going to talk about now what's called thermoregulation. Thermoregulation. This is the way that our body deals with I'm too hot or I'm too cold. Thermoregulation. Before we dive into this big picture, I'm going to take a step back and we're going to look at this one. This shows me the typical setup for what we call homeostasis. Homeostasis. There are a lot of different things in your body that we have to keep in balance or that we have to keep level. What are some of the things that you can think of that your body wants to keep at the correct level? Or put another way, what are some of the things that your doctor checks to see if you're at homeostasis, to see if you're balanced? What kind of things do we maintain the level of in our bodies? Yeah, so Max honing in on what we just saw in the other picture, body temperature, want to keep our temperature stable. Hayden's mentioning salt, absolutely, along with glucose, yeah, oxygen, levels of different kinds of electrolytes, or Caitlin's chiming in about some hormones. Yeah, almost everything in your body, pretty much everything in your body, we want to keep it at balance. We want it to be at homeostasis. But because we don't all live in a little box where nothing ever changes, we have to have these systems in place for when I'm out of balance to get back into balance. That's what homeostasis is. Something's out of balance. Here's how I get back to balance. So let's use body temperature, since we're going to dive into that more in just a bit. When we talk about body temperature, you've got your normal temperature, 98.6 or 37 degrees Celsius. Say, because it's Texas, I walk outside. And I walk outside and it's hot outside, because why wouldn't it be, right, in March? So I walk outside, it's hot, and the stimulus is the heat in the environment. That stimulus affects my body and leads my body temperature to go up. I'm now out of balance. I'm not where I'm supposed to be. Well, we have receptors in our body, special cells, that are constantly monitoring what our body temperature is. They want to know, are we at balance? Are we too high? Are we too low? My receptors detect what's going on with the temperature. If they detect that something's wrong, they don't know what to do to fix it, but they do know that if they send that information to the control center, which is typically in the brain, that control center can figure out what needs to be done. And once it figures out what needs to be done, it's going to talk to the effectors. The effectors are the part of things that get stuff done. So from detecting a change to predicting how to fix it to actually fixing it. That's the way the process of homeostasis works. So anything in your body that we keep at a balanced level, we have receptors and a control center and effectors that make sure it stays that way. Let's go back then to temperature. We talked about how temperature, we've got our normal balance of temperature. But there's two ways that our body can go. There's two different stimuli we can have related to temperature. We can have cold or we can have hot. These are my two stimuli. Now, I suspect that we can come up with the word that goes over here. What word would we use to describe when your body gets too cold or when your temperature is much lower than it should be? What would be the word for low temperature? Exactly. Yep, the word for a low body temperature is hypothermia. Hypothermia, just like hypotonic, where we had not enough salt. Hypotonic. Hypothermia is when my body temperature is too cold. Based on that hypo part, what do we think we would call it if our temperature was too high? If hypothermia is too low, what do we think we'd call it if the temperature was too high? Yeah, exactly. Hyperthermia. Hyper, too high. Hypo, too low. Either of them, we're not at balance. We're not where we should be. So let's talk the hyperthermia side of things first, over here on the left, and then we'll talk about the hypothermia side of things over here. So we're starting here at balance. We're starting at your normal body temperature. Again, we go outside in Texas heat, our body temperature goes up. It's not normal anymore. This is detected by a set of cells that live in your skin and they live in your brain. And their purpose in life is to constantly take your temperature. The cells on your skin, taking the temperature of the outside and the inside and mixing that information together. The cells in your brain are taking the temperature of the blood in your body. So if the temperature that we detect on your skin and the temperature we detect in your brain is higher than it should be, these receptor cells will send that message up to the brain. So we have a part in the brain that its one job in life is to control your temperature. It's called the thermoregulatory center. The thermoregulatory center gets the information from the receptors. The receptors said, we are too hot. The control center says, okay, I can fix this. So the first thing the control center is going to do is it's going to make you sweat. Sweating that watery sweat that helps to cool us off. Hopefully you remember this, especially my Wednesday lab friends. What's the name of the kind of gland that makes watery sweat? Watery sweat that would cool you off. What's that watery sweat gland? Yeah, the watery sweat gland is the eccrine glands. So when you get really hot, we sweat because when water comes off of your skin, it takes with it the heat from inside of you. So the first thing we do when we have hyperthermia, we activate, one of my effectors is the eccrine glands. They spit out sweat, that helps to cool me off. But the other thing that we do is we talk to our blood vessels. Remember those blood vessels we had in the dermal papillae? They helped to bring oxygen and nutrients to the skin. When I'm really hot, the control center says, hey, blood vessels, I need you to do vasodilation. I'm going to type it in the chat for us here. Vasodilation. Vaso means blood vessels. Dilation means making them bigger. Vasodilation means that more blood will get sent to your skin, and when the blood is close to your skin, it can radiate off its heat into the environment. So when I'm too hot, the two ways I'm going to lose heat. Number one, watery sweat from the eccrine glands. Number two, vasodilation, where I make my blood vessels bigger. I dilate them. If I'm cold, I do not want to be sending as much blood as possible to my skin to lose all of my heat. When I'm cold, my receptors, the same receptors that were detecting that I was hot before, now they detect that I'm cold. They still don't know what to do about it, but they send the information to the control center in the brain. And this time the control center says, oh, we're not too hot, we're too cold. I can fix this. So the first effector that we use when we have hypothermia is we activate the type of muscle tissue in your body that we said helps you with movement, with posture, and heat production. Do you remember which of our types of muscle tissue did movement, posture, and heat production? Which kind was that? Exactly. Yep, that's the skeletal muscle tissue. The first thing that we activate when we have hypothermia is our skeletal muscle tissue, causing us to shiver. When we shiver, our muscles contract, and that makes heat for us. So the first thing I'm going to do when I'm too cold is I'm going to start shivering. That's going to generate some heat. The other thing I'm going to do when I'm too cold is I'm going to talk to my blood vessels that I have in the skin, and I'm going to tell those blood vessels, hey, you need to get smaller. You need to do vasoconstriction. Vasoconstriction. So when I tell my blood vessels to get smaller, then I send less blood to the surface of the skin, which means I'm going to lose less heat there. When you're thinking about symptoms of hypothermia, remember that they talk about things like blue lips or blue fingers or toes. The reason those things are blue is because of vasoconstriction. When I constrict those blood vessels, I stop having as much blood going there, and they start to change color because we don't have this oxygen-rich blood going to them. The reason we're doing vasoconstriction, though, in places like your fingers and your toes, is because we're taking all of that blood that would have been going out here, and we're shuttling it into the thoracic and abdominal pelvic cavities. When we put it all there, we're protecting our organs. For our organs to work, they use a bunch of those proteins called enzymes. Remember, enzymes, a type of protein, get stuff done. If I get too hot or too cold, I might permanently lose my shape and also lose my function. What was that called when a protein permanently loses its shape and permanently loses its function? What would we say that protein is? Exactly. Yep. When I permanently lose my shape and my function, I am denatured. Remember that temperature has the ability to denature proteins. So when my body is cooling down, or for that matter, when body's getting too hot, the reason I'm doing homeostasis to keep at the right temperature is I don't want to denature my proteins. I don't want them to get so cold that they can't work anymore, and now I can't digest anything, or I can't clean my blood. I don't want them to get so hot that I can't do those things either. We maintain homeostasis, a steady temperature, because we need to protect the way that our organs work in our body. When we look at the way that body temperature regulation works, it's something that we call a negative feedback loop. A negative feedback loop. What that means is whatever my stimulus was, my effectors are going to do the opposite of that. So if I get hot, the effectors I'm going to activate are going to make me cool. If I got cold, the effectors that I activate are going to make my body temperature go up. In a negative feedback loop, we've got a normal, and that normal is where we always want to be. We don't want to change it. If we're changing, my body will return us back to that, a negative feedback loop. So almost everything in the body is going to be regulated by these negative feedback loops. The goal, keeping things steady. If we go too high, if we go too low, we can use a negative feedback loop to get back to normal. Let me check out my chat. Yeah, so Lisa's asking some interesting questions that relate to things like stimulants or withdrawal symptoms. So I'm not an expert on these things, but what I will say is anything that we put into our body has the ability to interact with all of our cells, especially all of our cells in the nervous system. So if I put a stimulant into my system, Dr. Alice will admit to this one. When I do my morning coffee, I get a little bit flushed. My face gets a little bit flushed because that caffeine is causing my blood vessels to dilate. I can't tell you for sure on a chemical level how that works, but what I will say is us changing the shape of our blood vessels, dilating them or constricting them, there are so many different ways that we can do that. We're doing it all the time to maintain your blood pressure. So if your blood pressure is really high, your blood vessels might be really constricted. Or if your blood pressure is too low, they might be really eased out. So there's lots of homeostasis loops all happening in your body at the same time. Throw in something like a stimulant, and you may not be able to maintain homeostasis at all. But yeah, great question. Any other questions about a negative feedback loop, like we see here in our picture? I've talked so much. Are you all still tracking with me? Can I get a thumbs up or an emoji? Are we halfway tracking, at least, with Dr. Alice? Okay, good. We're still tracking with me. Well, here's what I'll mention. In our next lecture, next week when we're in person, we're going to look at another picture that looks a lot like this, but it's going to be talking about the way your body maintains calcium levels. So this particular chart is showing me keeping balance in my body temperature, but I could trade out the balance being temperature and I could put it being a balance of calcium. So if I have too much calcium in my bloodstream or too little calcium in my bloodstream, that's going to cause me either to build some bone or to break down some bone. So we're going to come back to this again next week when we talk about the skeletal system. So keep that in mind. Negative feedback loops are not going away. They are here to stay. There is one other kind of feedback loop that your body uses, though not nearly as regularly. And this is what we call a positive feedback loop. Positive feedback loop. When we talk about a positive feedback loop, the best way I can think of to describe them is instead of going back to our original normal, what we were maintaining before, we actually make this new normal, and we do the new normal for a while until we've accomplished some purpose, and then we stop. So let's look at an example of a positive feedback loop, and that's the process of blood clotting. So say we got a paper cut, and the blood vessel in our finger has been cut. We're bleeding. We don't want to bleed out from a paper cut. So when we damage our blood vessel wall, that calls over some cells called platelets. Platelets are a type of cell that when they get damaged, they explode. They spit out all these proteins that form big, long lines to build me a blood clot. The other thing that these proteins that they spit out do, in addition to helping me make a blood clot, the other thing they do is they recruit more platelet friends. So these platelets were on the scene when the damage happened. They explode. They start to build proteins. Well, that explosion causes a bunch more platelets to come over, and they explode. And their explosion causes more platelets to come over, and they explode. And we go on and on and on in this cycle until we've built a clot to close up that blood vessel. Can we think of a reason why it might not be good for this to be happening forever? Why might it not be good for platelets to keep coming over and exploding for all time, forever and ever? Any ideas? Yeah, so Victor put forth an excellent idea. If we keep doing this forever and ever and ever, we might block the whole blood vessel. And in fact, that's what happens in a stroke. In a stroke, we somehow get some kind of clot that blocks a blood vessel in the brain. And when we block that blood vessel, no oxygen and no nutrients going there. So I don't want to do this positive feedback loop forever. I don't want to completely fill my blood vessel. I want to do the process of blood clotting for quite a while until I get to the point that my blood vessel is fixed. So again, in a positive feedback loop, something happens. We tore our blood vessel, and that caused some platelets to start clotting, which made others come over to clot, which made others come over to clot, and we go and go until there's no more damage in the blood vessel. Hey, another example of a positive feedback loop is the process of childbirth. In the process of childbirth, there's this interaction between the position of the baby in the uterus and what kind of messages the brain is sending to the uterus. So when it's close to the time for a woman to have a baby, her baby drops down in the uterus and starts to put pressure on the bottom of it. That pressure that the baby is putting on the bottom of the uterus sends a message up to the brain. The brain says, oh, the baby is down. Sends a message back to the uterus, contract. When the uterus contracts, it pushes the baby down, which sends a message to the brain. The brain sends a message back, contract. So we get this cycle where the brain keeps telling the uterus to contract, and the message from the uterus goes up to the brain, and the brain says, we're right on track. We're doing what we're supposed to. This goes on and on and on until what? What's my new normal? When do the contractions stop? Do I have any mothers in the class? When do the contractions finally stop? Oh man, we really don't know, or we're just afraid. Exactly. Contractions stop when the baby's gone. So the uterus will keep contracting. The brain will keep feeling that, oh, the baby is low. It keeps going and going until the baby is out of the uterus. Once the baby's out of the uterus, that pressure is no longer there, which means the brain is no longer going to send the message to contract. So we had a loop where they were constantly talking to each other, the brain and the uterus. But once we no longer need to do contractions, we won't anymore. So positive feedback loops not nearly as common in the body as negative feedback loops. Because again, in a positive feedback loop, we basically make a new normal. That new normal accomplishes a purpose for me. It builds a blood clot, it pushes out a baby. But once we've done those things, we're going to go back to normal. Positive feedback loops, negative feedback loops. The two big picture ways that pretty much everything in your body works comes down to these loops. Yes, so Lisa mentioned that she only has a theory about the process of childbirth, not experience. That's totally fine. Your theory was correct, that once that baby is birthed, as Hayden said, once that baby is out, we are done with contractions. So absolutely correct. So Cassidy asked a question about the afterbirth. I'm going to be honest, I'm not an expert on the birthing process. I believe they continue for just a little bit because the placenta needs to be delivered as well. And I do know as well that there are other factors that can lead the uterus to contract later too, like the hormones that the body produces when a mother is breastfeeding. That can also lead to contractions. So remember how before we were talking about when your body temperature goes crazy because of drugs, for example, in your system? It's the same thing with hormones. So hormones can influence our homeostasis loops as well. So cause us to kick back into a contraction cycle for a little while until those messages are done. But yeah, great question. Any other questions about positive and negative feedback loops? The way we keep things stable, right? Flat and stable. I will mention, I do not have it in my slides, but at the bottom of your lesson number 7 packet at the very end, there are several review questions that you see there. Even though we're not going to do them together here in class, I strongly recommend them for you as you're wrapping up your studying, as you're preparing for Monday's class. Monday's class, we don't have any lab quizzes. We just took the lab exam, but we do have a lecture quiz. So make sure as you're working on the lesson number 7 homework assignment, I've talked to a lot of people about this. Not only do we want to get our highest possible score on that assignment using our notes, we need to put those notes away and try to do it again. Because when we take that quiz next week, we don't get to use our notes. Practice for that quiz by working on that homework assignment without your notes. Again, remember, I only see your highest scoring attempt. So if you do really bad the first time with no notes, I don't care. I'm seeing your highest score from when you were using the notes. But a good way to practice, a good way to prepare would be to use those assignments like they're a quiz. That's some good practice to get you ready for those quizzes. So as we're closing up here, clarifying question, positive feedback is childbirth. Yeah, when we talk about negative feedback, exactly. Yep. So like Jessica mentioned, the whole word that we use for it, we call it thermoregulation. So whether we are shivering, whether we're sweating, that's an example. Both of those are involved in thermoregulation. So regulating your body temperature. But again, it's not just body temperature. It's all that stuff that we talked about, right? It's blood sugar, it's blood calcium, it's blood pressure. Almost everything in our body is a negative feedback loop. Not childbirth, not blood clotting, and the way that we talked a little bit about the thing called an action potential, where a message travels down my neurons to go from one cell to another. That's one other example of a positive feedback loop. But I'm probably not going to ask you about that one since we haven't covered it in depth yet in our lecture class. All right, well, let me turn off my recording.